Um, a very good evening to all participants. Welcome to this week 2 NPTEL live TA session for the course Cell Culture Technologies. I'm your TA for this session. My name is Ankita De, and I host this session every week on Tuesdays. So, as already discussed in the previous session, here we will be solving some of the previous year's assignment questions from the year 2021, and in the end we'll also provide you with the access to necessary PowerPoint slides and the recorded videos. Towards the end of the session, we'll also have a Q&A segment where you can ask me your questions. For any query, kindly use the chat box option and I will try to answer them. So let's get started. Now, before we start discussing the assignment questions, let's quickly have a look at what we have discussed in the previous lecture. So we have discussed what cell culture refers to and how it is done, and its various applications in the field of modern molecular biology research. Further, we have also talked about some of the keywords that are used in connection to cell culture, often such as a cell line, cell strain, uh, primary versus secondary culture, or finite versus continuous culture, adherent versus non-adherent cells, dividing versus non-dividing cells, and so on. So we have also tried to shed some light on the process of cell cycle division and have come across some critical terms such as differentiation, de-differentiation, and potency. Thereafter, we have solved some questions relating to that. And for those of you who haven't been able to attend the previous session, can refer to the recordings that have been made available to you on YouTube. Moving on to this week's, that is the week 2 NPTEL video lectures. So in this week's NPTEL video lecture for the course Cell Culture Technologies, Professor Moin Akhtar has primarily discussed about the layout of a cell culture room or facility and its necessary components and each of the uses. So further, we have also seen the importance of following aseptic techniques while working or before entering the cell culture room, uh, and some precautionary methods that we must keep in mind while designing the cell culture lab. So let's quickly have a look at each of these aspects. So the cell culture facility or room is usually set out somewhat in this way. It is an enclosed space which is separated from the rest of the lab, and entry and exit to and from this room is marked by sometimes one or sometimes more than one separate doors. So the main door which leads to the room is guarded by a wind curtain, which is like a vertical flow of air which prevents the entry of outside air into the cell culture room. So this main door shall usually lead to one room, which in turn has another door which will lead into the core cell culture facility. So outside the core, that is the core cell culture room, we have uh, several components where you can this is the place where you can put on your proper attire which is required while working in a cell culture lab, that is your lab coat, your goggles, the head cap, the gloves, etc. Also, you need to wash your hands before entering the core cell culture room. So before entering the core facility or the cell culture room, we place sticky mats at the door, which will usually attract any dust particles that might be drawn into the room along with the workers' feet. So and also this entry to this room, that is the two doors which will lead into your core cell culture room. These two doors must not be opened simultaneously. Only when one door, that is the main door, closes, shall you open the next door, which will actually lead you into the cell culture, the main cell culture room. Now, after we enter the core cell culture room, we need to have proper designated areas, somewhat like this. for each of the components, the necessary components, and each of them must be placed in an order to make efficient utilization of the space and maximize the effective work area, as well as prevent the culture space from coming in direct contact with outside air. So now we find inside the cell culture room, we find these important components, and one of them is this biosafety cabinets. So depending upon the number of people that are working in a lab, you can have from 1 to 2 biosafety cabinets. These are also referred to as the cell culture hood or the laminar flow hood, and they look like these enclosed box like structures with glass walls and a regulated air circulation system, which will provide us with a work surface or area to perform all our cell culture activities without without getting the risk of contamination from outside air. Now uh, so now we also the next important thing that we have inside our cell culture room are the carbon dioxide incubators. So moving on, this is our biosafety cabinet, and then right next to it we have our carbon dioxide incubator attached to CO2 cylinders. So the layout, depending on the layout of your lab, you can place them in like you can depending on the how your lab looks like, or how what kind of space you have available, you can place them accordingly. It's not necessary like you should place them right next to each other as is shown in this picture. So you what we have is the next we have the carbon dioxide incubator. So inside the carbon dioxide incubator we keep the cells under, we try to maintain the cells and in a physiological condition, that is we like proper, we uh, we provide it with proper humidity, temperature, and carbon dioxide concentration. So as you know already, as we have discussed in the previous class, that the cells require to be kept at 37 degree centigrade temperature and a constant supply of carbon dioxide, which is usually at around 5%. And this carbon dioxide which is supplied helps to act as a buffering system by combining with the water inside the cells and producing bicarbonate acid, and help to maintain the physiological level of pH, which is at around 7.4. Then we have the freezers. We have different freezers at different temperatures, some of which are say at 4 degrees centigrade. So the 4 degree centigrade freezer or the fridge is usually used to keep the cell culture media which we use for our cell culture. Then we have the minus 20 degree freezer, which is used to keep the necessary chemicals and reagents and some of the drugs maybe. And also we have the minus 80 degree freezer, which is used for short term storage of cell or protein extract before shifting them to the liquid nitrogen storage tanks. Then we also have a water bath, such as this one, at which is maintained at around 37 degrees centigrade. And then we have an inverted microscope, which is used to visualize the cell size, the cell number, density, morphology, or any possible contamination that might be arising in your cell culture plates. And then we have a cell culture centrifuge, which is used to which is used to pellet down the cells, which you are getting after suspension. That is, you are getting after detaching the cells from the culture plate, and you trypsinize them, and you get a cell suspension by flooding it with media, and then in order to pellet that cells, you have to use the cell culture centrifuge. Now after this we have the liquid nitrogen tanks. The liquid nitrogen tanks are used for the long-term storage of cells for years, and is maintained at below minus 130 degree centigrade. So inside this liquid nitrogen tanks, we have these kind of racks where, and inside each rack, you will place the cells in this way in liquid nitrogen in boxes which you can keep in at which are boxes which are able to endure such low temperatures of the liquid nitrogen. Then we have a hemocytometer. So which is a counting chamber or device which is used to measure, which is used to count the number of viable cells that you have. So at our lab we have an automated cell counter. We do not use a hemocytometer. This was used previously, but now we have a automated cell counter. So what we do is, if you put the cells on a slide, like you say you take 10 microliter of a cell suspension, you take 10 microliter of a dye, which is known as trypan blue. So, and you mix it together and you put it onto the surface of a slide and cover it with a cover slip, and then you insert it here. So then it will automatically give you the total number of cells and the number of dead cells and the number of live cells. So this is automated. You don't have to manually count the number of cells. So at our lab we use this, and the principle behind this is the trypan blue, the dye that we are using will usually, what it will do is, dead cells, their cell membrane is not intact, and so this dye will enter into only those cells whose cell membrane is not intact, that is the dead cells. So in that way we'll able to differentiate the live cells from the dead cells, and it will give you a count of the number of cells. So, and here we also have the carbon dioxide cylinders, as we have mentioned previously. So the carbon dioxide cylinders are connected to the CO2 incubators that we have. And then we have the culture vessels. So they come in different shapes and volumes. So we have the flask, cell culture flask, and then we have the cell culture plates, and then we have the 96 well plate, and then we have the 6 well plate, we have the 24 well plate, and then we have other glassware, pipette that we use, other consumables, like the pipette. We have different range of pipettes from say 10 ml to 1 microliter. So there are a range of pipettes that we use in our lab. Now moving on, as we were discussing the need for CO2 incubators in the cell culture facility. So as I've already discussed that the cells require a proper physiological conditions to grow outside the body, that is in an artificial environment. So we need to supply it with the appropriate culture conditions, like it needs to have a pH of 7.4 and a temperature of 37 degrees centigrade and a relative humidity of about 90%. So this is the way how a CO2 incubator works. So we supply the cells with a 5% CO2 concentration. This CO2 from the air will be will combine with the water present inside the cell to produce bicarbonic acid, H2CO3, and this will act as a buffer. This H plus ion will act as a buffer. Also, the medium in which we are culturing the cells have a pre-added NaHCO3, that is sodium bicarbonate. So together they will act as a buffer system. So as you know that as cells grow, say, and they respire, they will produce certain waste materials into the medium. So what we do for this purpose is we add a certain indicator to the media that we are using, which is a say phenol red media. So the phenol red media is gives us a bright pink color when it is in an alkaline condition. However, it turns to orangish and then a yellow color as the medium turns acidic. So as the cells will respire, as it will grow, it will release lactic acid into the medium. So this lactic acid will change the color of the medium to yellowish color. So at that time you will know that this is the time when you need to change the media. And say if your cells are already confluent by then, so if say your cells are not confluent, so then you need to change the media only. But say both the color of your media has changed to yellow, and also you are seeing that the entire surface area, that is the entire surface area of the culture plate, is covered with cells, then you also need to passage the cells. So this is how you will know when to do that. Now besides that, also if there is some source of contamination in your cell culture, then also the media will change color to yellow. So this is one way to detect that. Now moving on, so we were discussing about the importance of aseptic techniques. So we will discuss now what are the various aseptic techniques that we need to follow in the lab. So basically Aseptic techniques refer to a set of procedures that help to reduce the probability of contamination by microorganisms such as bacteria, fungi, and viruses, and etc. So the sources of contamination could be non-sterile consumables or the culture vessels that you're using, pipette tips which are not sterilized, media or the reagents, or the use of faulty incubators or unclean work surfaces. So to do that, what we need to have is a follow the aseptic techniques, which will ensure that there is minimum contamination, or there is no contamination from outside sources. So for that we need to follow a set of rules. For so First things, you need to examine your cells, as I've already mentioned. You need to examine your cells daily. That is, if you're seeing any change in the medium that you're using, is if there is any change in the color of the medium, or you can see if there is any turbidity in the medium. So these could be result of contamination in your cell culture. Also, what we need to do is we need to ensure that our work surface, that is on the surface of the biosafety cabinet that we are using, is entirely clean. And for that, what we need to do is that we need to place our biosafety cabinet as far away from the door as possible, to ensure that it's not close to the door, and it's not prone to the outside air that is coming inside, and not prone to contamination. So also, the work surface that you're using must be entirely cleaned with the help of 70% alcohol before and after each use, and the work surface must be kept as decluttered as possible, so that you have a larger area, say, to work with. And also, you must while working in the lab, that is before each use, you must sterilize the cell culture, the biosafety cabinet, with a UV light. So this UV light, usually use UVC lamp inside our cell culture, that is cell culture hood, which ensures that the air inside the hood is sterilized, and it will kill any germs, or any say the any contamination that might be arising. So this UV light before each use, it is kept on for about 15 minutes, and usually at the beginning of the day, when we say, suppose when we are starting with our work, we keep the UV on for 30 minutes, and for the rest of the day, before each use, we keep the UV on for about 15 minutes. And then after it's done, that is after 15 minutes, before you start working inside the cell culture hood, you will switch off the UV, because as you know that UV light is harmful to our skin. It might cause cancer. So next, what we need to do is we need to avoid spillage, that is spillage of media inside the hood. And if at all there is some kind of a spillage happening, we need to clean the surface with 70% alcohol, so that it does not lead to contamination. Also, one thing that we do in our lab is that we constantly, before using any pipette, say glass pipette, we properly flame the pipette before using. So this will also ensure that there is no contamination. But flaming is need not do flaming if you are using disposable pipettes. But if you are using glass pipettes that you will be reusing time and again, then you can flame this. That is, with the help of a Bunsen flame inside the hood, you can flame this. So the next thing that we need to follow is good personal hygiene. So, like I said, that before entering the lab, you must clean your hands with 70% alcohol, and then wipe before putting in any material into the cell culture hood. You need to wipe them properly with alcohol, and different pipettes, and the different say the materials that you're putting inside the lab must be properly sterilized. So the commercially available culture vessels that we get are usually come in a sterilized form, and other things that we use in the lab, such as say, as I mentioned, that say your pipette can, which are filled with pipettes, and say the tips that you're using, pipette tips. So these are some of the materials that you need to autoclave. So autoclaving is another method of sterilizing your cell culture equipments. So but it acts like a steam sterilizer and is used to sterilize the, say, water, lab equipments, etc. So it uses steam under pressure to kill any kind of bacteria, viruses, and spores present in the equipment or the cell culture media. But there is a limitation to using the autoclave. That is, you can only use heat stable equipments for autoclaving, as the autoclave subjects them to a temperature of about 121 degrees centigrade and a pressure of 15-pound pressure for 30 minutes. So this is how it is usually done. So moving on to the questions, now we'll be discussing the questions that were that from the year 2021. So this is our first question. So you can answer in the chat box, and we will discuss what each of the right options are. So the first question is, one of the major roles of carbon dioxide in cell culture is to, and we have four options here, that is imbalanced pH, balanced pH, balanced surface tension, and imbalanced surface tension. So you can write in the chat box what you think the right answer is, and then we will discuss. Okay, so many of you are saying that it's the option B, that is balanced pH. Uh, yeah, so let's see what the right answer is. Yes, so the right answer is to balance pH, as we have discussed previously that carbon dioxide helps to act like a buffer system. It helps to maintain the pH to the physiological level of 7.4. So it's true that so this is the right option, that carbon dioxide in the cell culture is used to balance the pH. Now moving on to our next question. So the entry and exit of a lab should be clearly defined and not obstructed to ensure. Then we have four options: safe exit in case of emergency evacuation, safe entry of external air in the lab, aesthetic appeal of the lab, or nobody can enter the lab. So, so you can write the answers in the chat box again, and we will see and discuss each of the options. So a couple of you are saying that the right option should be option A, safe exit in case of emergency evacuation, and a couple of you are saying that it should be option C, that is aesthetic appeal of the lab. So let's discuss what the correct answer should be. So option A, that is safe exit in case of emergency evacuation. So as we might have seen in this week's video lectures, that Professor Moinak Das has already discussed about this aspect in the cell culture lab. That is, the entry to the cell culture lab, it's usually marked by two doors. It is one, the entry, and another exit. But usually this is not followed in all cell culture labs. Just like in our lab, we have a single exit. So if you have space, that is, if your lab is laid out in such a way that you can have two doors to it, then you can have. So it is always desirable that you have an emergency exit from the lab, so in case of emergency evacuation. So The first option is safe exit in case of emergency evacuation. Now let's talk about the second option, safe entry of external air in the lab. So we have already discussed that the cell culture room is uh, prepared in such a way that it will prevent the entry of outside air into the lab. So of course this is not the right option, that safe entry of external air in the lab, because our our aim is to prevent the entry of external air into the lab. The third option is the aesthetic appeal of the lab. So aesthetic appeal basically refers to the how your lab looks, that how decorated it looks, and all that. So the last thing you would consider while designing your lab is the aesthetic appeal. So what we need to ensure is that we need to design our lab in such a way so that there is minimum contamination from outside. So how it looks is not a concern. So basically what we need to ensure is that the cell culture room should prevent the entry of outside air, should prevent anything coming from outside into the inside. So it is not about the aesthetic appeal, it's about the utility. The next thing, the next option, the option fourth is of course nobody can enter the lab, which is somewhat of a very absurd option, and it's of course it doesn't look like the right option. So let's see what the right option should be. So the right option is option A, safe exit in case of emergency evacuation. Okay, so now let's move on to the next question. Aseptic techniques are employed in cell culture lab, and we have four options here: to contaminate cultures, to prevent contamination of cultures, to remove dust, or for the aesthetic appeal of the lab. So you can again write in the chat box what you feel is the right option. Okay, so many of you are saying that it should be option B, that is to prevent contamination of culture. So as we have already discussed that the aseptic techniques refer to a set of procedures which which ensure no contamination of the cell culture. So of course it is to prevent the contamination of culture, and so let's see what the right option is. So the right option is option B. So all of you who have written that option B is the right answer, that is true. So now let's move on to the next question. So which of these is used in the cell culture lab to store cell line vials for long term? Liquid oxygen, liquid carbon dioxide, liquid nitrogen, or liquid hydrogen? So again, you can use the chat box to answer, and we will see what the right option is. So all of you are saying that it should be option C, that is liquid nitrogen. So we have already discussed that liquid nitrogen has a temperature of minus 196 degrees centigrade, and for long term storage of our cells we need to store them at temperatures below minus 130 degrees centigrade. So while we are freezing cells, we first place them, we mix the cells in a freezing medium which will help to which will help to prevent the formation of ice crystals in the cell. So we mix our cells with an appropriate freezing medium, and then we place the cells for short term storage at minus 20 degree for about say 3 to 4 hours, and then we shift the cells to minus 80 degree centigrade. So this is how it is done in our lab. Other labs may have other procedures. So we from minus 20 degrees centigrade we shift the cells to minus 80 degrees centigrade, where we keep it for around 5 to 6 days, and then we shift the cells to the liquid nitrogen tanks. And in the liquid nitrogen tanks you can store the cells for a long time, say for years. So this and for at minus 80 degrees centigrade, you can usually store the cells for about roughly 6 months. Okay, so once you've frozen the cells, you need to shift it to minus 196 degrees centigrade, that is to the liquid nitrogen tanks eventually. So let's see what the right answer is. So the right answer is of course liquid nitrogen. Now let's move on to the next question. HEPA, that is HEPA, stands for High Efficiency Particulate Air Filter, High Efficiency Primed Animal, Health and Emergency Patient Accessibility, or High Efficiency Primary Flow. So you can write in the chat box. So the HEPA or HEPA filters are placed in our biosafety cabinets, and they have um these are placed inside the biosafety cabinet, which in the exhaust system, so that it will effectively trap all known infectious agents and ensure that only say microbe free air is discharged out of the hood. So what do you think the right answer is? We will discuss now. So many of you are saying that it should be option A, that is high efficiency particulate filter. So let's see. I'm sorry, so there's a slide missing. So yes, the right option is high efficiency particulate air filter. So the answer slide has not been provided here due to some mistake. So now we will move on to the next question. So question number 6 is, which of the following is not safe to be autoclaved? Stainless steel, high density polyethylene, surgical equipments, or glass? So you can write in the chat box the, the right option, what you think. So as we have discussed that autoclave is a steam sterilization uh, procedure, steam sterilizer, which is used to sterilize our lab equipments, and that you can only autoclave those materials which are say heat stable. So the heat level equipments cannot be are not safe to be autoclaved. So the autoclave uses a temperature of 121 degrees centigrade at 15 pound pressure for 15 to 20 minutes. And that is enough to destroy any bacteria or viruses or spores that might be present in the equipment. So let's see what the right option should be. So the right option is high density polyethylene, because it is as I said, it is not high density polyethylene is a plastic ware. It's not safe to be uh, autoclaved. It might lead to melting of that, so it is not okay to autoclave it. Stainless steel, as I have already discussed, that your pipette cans are made up of stainless steel, so you they are safe to be autoclaved. And then we have the surgical equipments and glass equipments. So the right option is high density polyethylene. Now let's move on to the next question. So we have a blank and it says the properties of cells are required to relate the cultured cells to functional cells in the tissue from which they were derived. So this question is somewhat related to the previous uh, session that we had. So in that session we had discussed about these aspects of cells. We have four options here: dedifferentiation, apoptosis, differentiation, and aneuploid. So what properties of cells are required to relate the cultured cells to functional cells in the tissue from which they were derived? So let's see what the right option is and let's discuss each of the options. So differentiation, as we had discussed in our previous class, refers to the acquisition of specialized characters of a more progenitor cell. So progenitor cells, which is that is the cells which have the maximum potency to be able to differentiate into any cell inside our body. So these progenitor cells will gradually acquire more specialized characters and they will move towards a terminally differentiated state. That is, the terminally differentiated state refers to the state where the cell has acquired the maximum or the highest level of specialization. So, inside our tissue, there is any kind of specialized tissue that you might have in our body. So, suppose we are talking about here, previously in our previous class, we had discussed about the neurons. So, about the nervous system. So, the neurons are the terminally differentiated cells. So, they won't divide further because they are terminally differentiated because they have achieved the highest level of specialization. So, in order to relate the uh, cells, that is the cells, the tissue from which you are deriving the cells, so the tissue, usually what we do, we take out a chunk of tissue from outside the body and then we disaggregate the cells because a tissue refers to a group of cells which have a similar function and structure. So we disaggregate these cells and we make them into a single cells and then we culture them in a culture plate. So in order to relate the cultured cells to the functional cells in the tissue, what we need is that what properties of cells are required are the differentiation properties. Okay, so differentiation is the right answer here, and many of you have already answered. That's the option C, which is the correct option. And de-differentiation here is the exact opposite process that is happening. That is, de-differentiation is the loss of specialized characters of a specialized cell. So the right option is differentiation. Let's see. So yes, differentiation is the right option. So now let's move on to the next question. Using a pH meter, you find the pH of an unknown solution to be 8.0. How would you describe this solution? Strongly basic, weakly acidic, or strongly acidic? So again, we have four options. So as we were discussing that we add a certain indicator to our media, that is cell culture media. This indicator, which is phenol red, has gives us a different color at different pH. And with the help of this indicator, we can determine the pH of our cell culture media, the cell culture that we are using. So the phenol red indicator will usually give us a pH, will usually give us a color, bright pink color when it is at a alkaline pH, that is a basic pH. It will turn to yellow at an acidic pH. So using a pH meter, you find the pH of an unknown solution to be 8. So as you must be aware that 8 is, as you might be aware of the pH scale, that it has a 0 to 14. It starts at 0 and ends at 14. So anything beyond 7, that is 7 to 14, is the alkaline range, and below 7, that is 0 to 7, is the acidic range. So anything above 7 is 8, and since it's not very far away from 7, it must be weakly basic, right? So as you have, couple of you have already answered in the chat box that the option must be B. So let's see. Yes, so the right option is weakly basic. Now let's move on to the next question. Which light is used to sterilize the air in the laminar flow hood between uses. So we have four options: IR, NIR, UV, or visible. So IR refers to infrared, NIR refers to near infrared, UV is the ultraviolet light, and then we have the visible light. So which of these options is the correct option? Which light do we use to sterilize the air in the laminar flow hood between uses? So you can write in the chat box. So a few of you are already saying that it should be option C, UV. So as we have discussed that before that is before we start working in the cell culture lab, the beginning of the day, we switch on the UV light in our cell culture room, and also the UV light inside the cell culture hood. So we sterilize the air inside the cell culture room with the help of a UVC lamp, and between each use, at the beginning of the day, we keep the the UV switch on for about 30 minutes, and then after before each use, we keep it on for 15 minutes. This UV light must be switched off before we use the cell culture hood, because the UVC light is harmful for our skin. So let's see. So yes, the right option is option C, UV. Now let's move on to the last question, question number 10. A major goal in embryonic stem cell biology is to develop protocols that enable lineage specific control of, then we have a blank, and then we have four options: division, differentiation, survival, or all of the above. So you can write in the chat box what you feel is the right option. So this also we had discussed in our previous class. So we had discussed about the embryonic stem cells. So as we were discussing that the zygote, which is formed by the union of the male and the female gamete, will gradually divide to give rise to the modula and then a blastula. So the blastula is divided into two components. So outside it forms, we have the outer cell mass, and inside this we have the inner cell mass. The inner cell mass will give rise to the embryonic stem cells. The embryonic stem cells are also the pluripotent stem cells, because they are capable of giving rise to any kind of cell in our body except the extra embryonic tissue. And the outer cell mass will give rise to the trophoblast and the extra embryonic tissue. So uh, the pluripotent cells, the embryonic stem cells, find applications in regenerative medicine, such as organ repair or tissue repair, etc. Um, so you can write down which of the following do you feel is the right answer, and then we will see. So moving on, let's see what the right answer is. Yes. So many of you have already said that it's option D, all of the above. So the goal of the embryonic stem cells, or any stem cell, is that it has two essential features. A stem cell can divide to give rise to another cell of its own kind, and it can give rise to and can divide to give rise to another cell which is differentiated cell. So it can both give rise to a differentiated cell and another stem cell. So the main aspect of an embryonic stem cell is that it can divide and it can differentiate, and for survival. So the options are the three options, all of them are the right options, and so option D, all of the above, is the correct option. So that was the end of our uh, problem solving session. So before moving on, I would like to acknowledge NPTEL and IIT Madras for giving me this TAship opportunity. I would also like to thank Professor Mainak Das, the course instructor for this course, for selecting me for this role. And I would like to express my gratitude to my supervisor, Professor Mahitosh Mandal sir. I would also like to thank MHRD, the Government of India, for the Prime Minister's Research Fellowship, and IIT Kharagpur for allowing me to pursue PhD at the institution. Lastly, I would like to thank all of you for joining this session. Thank you. So now moving on, we can, if you have any queries, you can post in the chat box and I shall try to answer them. So one of you is asking that my link to the YouTube channel. So I think the channel link is provided by NPTEL, that is, they will separately mail you the link. So at this moment, I do not have the link, but okay, but I think NPTEL will provide you with that. So any more questions if we have? So I will stop the recording now.